Check. Hello. So few are, all are there. Few are also in virtually they are joining.
Virtually is only 3.
Hello.
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 Check. Hello. Good afternoon, everyone. Very warm welcome to 6th International Conference on Agriculture for Sustainable Development. Let us begin with the parallel session. I would request all of you to kindly come ahead and join us here. Let us begin with the first session, and let's welcome the chairpersons, Dr. S.D. Mahapatra, Head Crop Protection Division, ICAR-NRRI, Cuttack. And also with us we have Dr. Anna Malai M, eminent scientist, ICAR-NRRI, Cuttack. I request all of you to please welcome both the session chairs with a round of applause. And our first speaker, let's welcome her, Ipsita Priyadarshini Samal from CUTM Bhubaneswar. You may proceed with your session. Yes, please, sir.
Please give your program. Where is your program? How many speakers are there?
So actually in this program, 8 presentations is there. In that 5 is physical mode, 3 is virtual mode.
Also, we have another presenter.
Okay, that means 9.
9, yes, sir.
Okay, that means 6 physical, sir, and 3 virtual.
What is the theme of the session? Theme.
The theme of the session is crop protection and management, sir.
Actually, sir, in this program, what happened, the theme has not been segregated clearly, sir, actually. There is mixages of all
Different topics, right?
There is mixages of all the subjects, sir, actually. It is not only pertaining to crop protection.
Okay.
That is the issue.
So, very good afternoon. So, welcome to this session. So, this is the session. So, it is not virtually segregated, but we can call it as a crop protection session, biotic stress. So, there are around 7 speakers, young researcher. So, others, how many were in forum? Yeah, online?
Online 3 are there, and here 6.
6 is there, physical mode.
6 are in physical mode.
Yes, sir.
So, there will be 15 minutes for each speaker. So, out of 15 minutes, only 12 minutes will be given for presentation. So, we will be stick to 12 minutes and 3 minutes will be discussion. So, first speaker is Ipsita Priyadarshini Samal. She is from Centurion University. So, I request Ipsita to present. So, please be very brief, to the point, no need of any much. This is a research work. Okay. So, be very specific, no need of much introduction. Only what is the objective and what are the methods. If there is normal methods, no need of explanation more, and coming to the results. Okay. So, please.
Good afternoon, everyone. Myself, Ipsita Priyadarshini Samal, doing PhD under the supervision of Dr. Gyanranjan Malik, Associate Professor, Department of Botany, Centurion University of Technology and Management, Bhubaneswar. Today, I am going to deliver my presentation on the topic Amaranthus viridis and Lantana camara, a study of their allelopathic effect on seed germination and yearly crop growth. So these are my contents: introduction, need of the research, materials and methods, result and discussion, and conclusion. So coming to the introduction, first we have to know that what is allelopathy, as this term is new for somebody. Then what is allelopathy? Allelopathy is a biological phenomenon in which one organism or one plant produces some biochemical substances that influence the morphology and physiology of its nearby or its surrounding plants. This process involved in the transport of some chemicals, and those chemicals are known as allelochemicals, from one plant to another plant by various methods, and those methods are root excavation, leaching, deposition of leaf particles on the soil, and volatilization. And these allelochemicals are secondary metabolites which not required for the growth, development, and reproduction, and these are known as the biochemicals. These may have a negative impact on the target organism. And next, coming to what is weed, we know the undesirable plant which grow naturally along with the main crop are known as weeds, and weeds compete with other crops for nutrients, sunlight, water, space, and other sources. So we have to manage the weed, or the weed management is very important in crop field. From this figure, we can easily know about the allelopathic effect. Here, this is the donor part, and that is the recipient plant. Donor plant produces some biochemical which are known as allelochemicals by various methods like volatilization, leachate, residue. And these allelochemicals have a beneficial or maybe some harmful effect on recipient plant. In many research, it is found that allelochemicals or this allelopathy effect has harmful effect on other plants, and due to the allelochemicals released from the donor plant, the recipient plant and its growth suppresses, and that plant, that is Lantana camara and Amaranthus viridis, these two weeds I have taken for my experiment. As we know that this data is all about Amaranthus viridis. We know that Amaranthus viridis is a edible plant. We eat this plant in our daily life, but this plant is often considered as a weed due to its aggressive growth pattern, and this plant is also known as pig weed. It has several names, and these are some literature review or its data that Amaranthus viridis has also allelopathic effect, or it has some poisonous effect or some harmful effect on other plants. And then Lantana camara, from Lantana camara, which is a perennial shrub. The first Lantana camara was discovered in 1841 at Australia. Some people this Lantana camara was brought to India for ornamental purpose only, but after that this Lantana camara introduced to India in early 19th, but this species was spread across 2 lakh hectare in our India also. So this Lantana camara is also a very harmful weed for our crop field. So tea, coffee, sugarcane, cotton plantation along with millions of acres cultivated land have also all been impacted by this species Lantana camara. So its management is also required. So next, coming to need of this research is, as we know that weeds cause more yield loss in crop production other than pests. So synthetic herbicides have become widely employed to weed control. In our daily life, we use synthetic herbicides to control the weeds. However, widespread use of synthetic herbicide has effects on our human health and the environment. Because of numerous environment and health concerns linked with this synthetic herbicide, we can investigate alternate method for weed management, and this alternative method is allelopathic effect, or through allelopathic effect of weeds we can prepare some bioherbicide that can we use rather than this synthetic herbicide or synthetic weedicide. As we know, weeds are unwanted plants, and their overgrowth causes problem for us and our crop field. That's why we just cut this and threw. Rather than throwing these weeds, we can use these weeds for making natural herbicide, weedicide or pesticide. So these are the basic methods. First, we have to collect the plant materials. These weeds which are found at different areas like cultivated field, wasteland, roadside areas. And I have collected these plants from different areas like this, and the Microtyloma uniflorum that I have taken as the crop, as this plant grows very easily and in a short time period. That's why I choose Microtyloma uniflorum, and I have collected this plant from OUIT Bhubaneswar. Then we have to prepare the aqueous extract of weed, as we have to see many experiments regarding these weeds, what its effect, what is its effect on the crops. So we have to prepare the aqueous extract. For aqueous extract, first we have to collect the plant, then we have to separate the plant leaves from the plant, and then we have to sun dry this, then we have to powder the leaf, and the sample solution of various we have to prepare the sample solution with various concentration. Like I have taken here 10, 50, 100 and 200 mg per ml. This concentration I have taken, and I have taken the distilled water as the control. Then we have to see the seed germination assay and screening. Healthy and fresh seeds of Macrodilema uniflorum were kept on the moist filter paper, and these filter papers were soaked with different concentration of weed that I have taken 10, 20, 50, 100 and 200 mg. Then controls were treated with 20 ml of distilled water. The seeds were kept under the dark condition for 4 days in a room temperature. Then the germination was considered, and the radical length were measured when it is equivalent to 2 mm or more in length. Then the radical length was measured after 4 days of germination, and in this figure we can see that Macrodilema uniflorum were kept on the moist filter paper with different concentration of weed extract. Then we have to calculate the water content and biomass. First we have to measure the weight, the fresh weight, then we have to measure the dry weight. Then photosynthetic pigments also calculated here. Then as we know that in my result and discussion part, I found some allelopathic effect or some harmful effect on these weeds. That's why it is confirmed that there are some secondary metabolite or some phytoconstituent present in that weed. So these are some presence of phytoconstituents were done according to different protocols of different tests like alkaloid, carbohydrate. Then coming to the result and discussion part, here we can see that first is seed germination assay and plant growth. Here the rate of germination of Macrodilema uniflorum seeds respond differently manner to different concentration. The germination percentage was observed, that is in the control condition 85.55%, and the radical length decreased with increasing aqueous extract condition of weed extract, which we can find in the image. Here in the control condition, the seed germination is highest, and when the concentration increases, the seed germination level is decreases. Then here from the graphs we can found that this graph shows the germination percentage of Macrodilema uniflorum exposed to different concentration. In control condition, the germination percentage is highest, and when the concentration increases, the germination percentage decreases. And here the graph shows the radical length of Macrodilema uniflorum exposed to different concentration of wood extract. Then effect of weed on growth of Macrodilema uniflorum. From this image we can found that the root length and shoot length is highest in control condition, and when the concentration increases, the root length and shoot length decreases accordingly. And this graph shows that was the graph that shows the shoot and root length of Macrodilema uniflorum exposed to different concentration. Then effect of weeds and biomass of Macrodilema uniflorum. Here the same, we can found that the biomass is highest in control condition, and when the concentration level increases, the biomass calculation of biomass also decreases. And here the changes in photosynthetic pigment, we have calculated here the chlorophyll A, chlorophyll B, total chlorophyll and carotenoid. Here in all cases, in chlorophyll A, chlorophyll B, total chlorophyll and carotenoid, the in control condition all these are highest, and when the concentration increases, the level decreases. Here the presence of phytoconstituent, what I have found in Amaranthus viridis, and here these are the phytoconstituents present in the Lantana camara. So from this all total experiment, we have found that the rate of seed germination, plant growth, biochemical parameters of Macrodilema uniflorum increases in control condition as compared with plant exposed to different concentration of weed extract. There was a noticeable reduction in seed germination, plant growth, that is shoot and root length, photosynthetic pigment, protein content in plant treated with weed extract, while there was an increase in phenolic, but I could not put here the data. The increase in phenolic content might be decreased due to the inherent capacity. In this present study, the weed extract inhibit the crop plant's growth and physiology. This whole study will definitely lead to the identification of few phytochemical or few plant metabolites, secondary metabolites having beneficial agricultural application for sustainability in near future. Sensitivity to the allelochemicals vary from crop to crop, but it will enhance future aspects of crop protection from several harmful plant species. Thank you.
So Now the topic is open for discussion. Any question from the audience? Why you have chosen this horse gram? What is the reason?
Sir, the reason this horse gram plant is easily grown in a short time of period. For my experiment, actually, I have to work this experiment on also rice, black gram and green gram. But this horse gram is very easy to grow, and in a short time of period, it can grow. That's why I have taken this.
So what will be your recommendation, particularly this lantana camera and this amaranthus?
From all these experiments, we have found that lantana camara and amaranthus viridis both have allelopathic effect, and when we used the synthetic herbicide or synthetic weedicide to kill the weeds rather than the synthetic herbicide, as these have allelopathic effect and these can kill all the weeds. That's why we can prefer all this, and we can make bio herbicide or bio weedicide. It has some effect, maybe some effect from review of literature. I have found that these plants have also some effect on insect or some pest. Then in future we can also make some bio pesticide from all these plants.
Why is it the new, whether earlier this effect was not known?
Sir, it's a secondary metabolite, and it's the secondary metabolite present in it, and the some of the allelopathic effect like from review of literature I have known that the surrounding neighboring plants surrounding these plants are affected by these plants. So from review of literature I have confirmed that this plant have some allelopathic effect. That's why I have chosen this plant, and in future I have to carry on my experiment so that we can make some bioherbicide or bioweedicide from it.
Okay. So have you encountered any, because this is one weed?
Yes, sir.
Under lantana camera.
Yes, sir.
And you know amaranthus is a very, though it is underutilized crop,
Yes, sir.
but it has very good value.
Yes, sir.
In northern India, they use this as a, who are going for the fasting,
Yes, sir.
they used to take this one.
Sir, Amaranthus viridis, when it is in a fresh condition, it is healthy for us. But from some experiment, it was found that when the plant is dry, it has some oxalate present here, which causes kidney problem also. So it is considered as a poisonous weeds also when the plant is dry. That's why we have to collect, or we have to make the extract by drying the plant, and then we have to make the weedicide.
After drying only.
Yes, sir.
But it is always used in Odisha condition as a vegetable crop.
Yes, sir.
In maturity conditions after drying, so that grains, that is also used for laddu, preparation of laddu, then preparation of the food grains. So how we can make it as a weed?
So from a review of literature and
Review.
some research also, sir, it is found that it is considered as a poisonous weed.
So first of all, let me clarify,
Yes, sir.
or you can educate me. So what exactly weed is
Sir,
in
actually,
your perception?
weed is an unwanted plant
Hmm.
which we do not grow or we do not cultivate a weed. Weed grows naturally. So in this Amaranthus viridis and Lantana camara, we cultivate it for our agriculture purpose or our fooding purpose. But in some cases, this Amaranthus viridis or Lantana camara, we don't cultivate it; it grows automatically. That's why it is considered as a weed also, sir.
So many cases are now medicinal plants and
Yes.
many herbs and herbs are there.
Yes,
They
sir.
cannot grow that herbs and medicinal plants. They
Yes, sir.
automatically grow. So they are considered as a weed.
Sorry, sir. Again.
Suppose in the medicinal plants,
Yes, sir.
and many plants that have some medical, some herbs and herbs are
Yes, sir.
there. So So they are automatically in the wild form. They are available. They are automatically grown.
Yes, sir. These are forms under the weed group, but those weeds have some medicinal plants. There are
As per
many.
your definition, that should be comes under the weed category or the
It's a weed category, but we can say that these weeds have some medicinal properties.
So do you know one Bathua?
Yes, sir.
So Bathua, one variety is really in a other condition it is a weed, but in some other condition it is a green. Pusa Bathua one variety has been released. So what Doctor asked, you can supplement that in the context, in the situation that may be considered as weed, and in some other context that may be considered as the crop or medicinal plants or the food grains. Okay, thank you, Ipsita.
Just to
Even-
probably one query. In your conclusion, you told it also affects crop growth and phenology. Whether it won't affect the main crop when you are going for having a phytocompounds from the plant, it won't affect the main crop. Then now you can recommend it.
No, sir. For that purpose, for that experiment, first we have to see that either it has some.
Okay, thank you. Next speaker is Priyanka Raha. All the speakers are requested to upload their presentation.
Better you complete the presentation upload. Swagatika, upload completely. Better we will complete all the physical. And then virtual, all the three we will keep on one stage.
Let them be in the line
I
online
am telling because they have to present in this
session.
session. It is not only that they can come on their time and present and go back. They also want to know what is happening. At least they have to gain something. Just only for presentation purpose they are coming means.
Yeah, I'm there only. But 121, I am leaving earlier now.
That's no issue. That's
Oh, you
I is
are
there.
you are leaving.
Yeah, leaving on 21.
no
Okay.
issue. So that's no issue.
So you are from IRI. Which division? Water Tank. Dr. Brahmanand is there.
Yes, our PD. Very nice.
ये चीज पहले से कर देना चाहिए ना. stress है. So actually we are running short of time, so we curtail the timing. So it be very specific to 10 minutes. So within 8 minutes you have to complete your presentation and 2 minutes will be discussion. So because at 5 o'clock we have the inauguration again.
Good afternoon to the respected chairperson and to other dignitaries. Today I am presenting a part of my research work on micropropagation of Limonium misty blue based on in vitro floral reversion and their morphohistological studies. Suddenly we aware the goals of SDG and already that has been discussed throughout the seminar. According to, day by day, the sudden climatic change leads to higher temperature and dumpy pattern of rains, which increase the problem of soil salinity day by day. Globally, in total, there are more than 833 million hectares of soil already affected by the salt, and which is 8.7% of the total planet. According to Mass 1990, less than 2 decibel per meter salt resulted in crop yield is a reduction. So one of the option is saline agriculture. On this context, what is saline agriculture? I am coming to the next topic. In 2019, the International Saline Future Conference fixed up a goal to identify and improve salt-tolerant plant variety having commercial interest. In this context, we have identified limonium, which have a Mediterranean origin and also is a recruitohalophytes, can even grow more than 500 millimolar salt and do not show any effect on stem yield and length if irrigated with the water with its EC up to 11.5 decibel per meter. So what is the importance of the limonium plant? First of all, limonium plant can grow on the soil, saline soil as well as the drought condition, and other portion it produces a marvelous eye-catchy purple blue color flower, which is popularly used as the cut flower and mainly used as filler in floral decoration. And other part, the limonium price is more than $17. That is one bunch of limonium plant that's contain 1 to 10 stick. And limonium produced by the renowned organization of our India, that is KF Bioplant, associated with the Hilberda florist, and supplying to the growers for the cultivation. So in this respect, we have devised an efficient protocol of shoot regeneration from thin cell layer section of inflorescence node indirectly based on floral reversion of Limonium misty blue, a hybrid plant between Limonium latifolium and Limonium bellidifolium, and finally transferred to the microplants to the field. And background study, there is only one report of Limonium misty blue micropropagation according to Toponoid et al., which reported that regeneration of 3 to 4 shoots from the inflorescence of the lateral node of the Limonium inflorescence